Here we are going to study the continuation of part 3 of vulva development. Here are some questions regarding some of the exam which you children need to discuss. The question number 1. Development of vulva in C. elegans is initiated by the induction of small number of cells by short signal from a single inducing cell. With reference to this, following statements were put forward. Number 1 statement. When the anchor cell was ablated early in the development, no vulva formed. B part. In a dominant negative mutant of Let23, a primary vulva formed, but the secondary vulva formation did not take place. Part C. A cell adopting a primary fate inhibits adjacent cells from adopting the same fate by lateral inhibition involving Lin39 and also induces the secondary fate in these cells. D part. A constitutive signal from the hypodermis inhibits the development of both the primary and the secondary fates, but it is overruled by the initial signal from the anchor cell. Question starts from here actually. Above the statement you have to go through nicely and never worry. Everything can be done when you approach with a happy mind. Which of the above statement is true? Here some numbers are given. Number 1, A and B statement. Number 2, A and C statement. Number 3, A and D statement. Number 4, B and D statement. Now you have to note, instead of Lin39 it should be Lin12. Ablated means removal. Here is the word ablated in the point A where it is underlined ablated early. It means removed. And here where the green color is on the Lin39, instead of this the word should be Lin12, which causes lateral inhibition. Here something is wrong. So we go through the answer. What should be the answer? As the question asks in the above line, with reference to this, following statements were put forward. So what is the answer? The answer is A and D, which is tick marked and with green color it is shown. Which of the above statement is true? Two statements are true in this and two statements are false in this. I have written T for true and F for false. Let us again go through it and see what is the actual problem in that. When the anchor cell was ablated, means removed early in the development, no vulva formation. Anchor cell is producing a ligand Lin3 which is activating VPCs as we learned in the previous part 3. B part, B statement. In a dominant negative mutant Let23, a primary vulva formed, but the secondary vulva formation did not take place. This is false statement, because instead of Let23, in a dominant negative mutant Let23 is not there only. How will it activate the VPCs? The C statement. A cell adopting a primary fate inhibits adjacent cells from adopting the same fate by lateral inhibition involving Lin39 and also induces the secondary fate in these cells. Instead of Lin39, here it should be Lin12. Then the fourth statement, which is D. The constitutive signal from the hypodermis inhibits the development of both primary and the secondary fates, but it is overruled by initial signal from the anchor cell. So this statement is also true. The answer comes A and D. Now going to the next question. During vulva development in C. elegans, the anchor cell produces Lin3 protein which interacts with the Let23 protein present in the 6 vulvar precursor cell that form an equivalence group. Thus, it is also called central lineage. The central lineage cell P6P adopts a primary fate. The adjacent VPCs P5P to P7P adopt the secondary fate and the rest VPCs adopt the tertiary fate. few mutants in the column A, here this one, and the few phenotypes column B are listed in the table to learn. This is the question which came in CSIR 2017 December. Now the question starts here. Column A, loss of function of lin3. It is also can be depicted as LOF, lin3. LOF means loss of function of 3. In front of it is, in the column B is written P5P, P6P and P7P adopt primary fate. P4P and P8P adopt secondary fate. Then moving further in the column A, the B part, loss of function of lin3 and gain of function of Let23 means LOF of lin3, GOF stands for gain of function, Let23. What will be formed? In the question, multivulva is written opposite to this, number second in front of the B part. Then C part is reduced function of lin3. In front of it is written P6P adopt primary fate and the rest of the VPCs adopt tertiary fate. D part, overexpression of lin3 causes all VPCs adopt a tertiary fate. It is written like this in the question. Now we have to match the correct mutant with the observed phenotype. Here examples are given. The answer is part A, where A in the column A, part A will be combining with fourth part. Loss of function of lin3, which is called LOF lin3, because lin3 is a ligand which is activating VPC, so all VPCs adopt tertiary fate when it is absent. Now B is second part. B part is loss of function of lin3 and gain of function of Let23, which will be combining with second part. Second part is multivulva will be formed. C part is reduced function of lin3. It is combining with part 3. Reduced function of lin3. Part 3 is P6P adopt primary fate and the rest of the VPCs adopt tertiary fate. This is the answer. Part D, overexpression of lin3, what it causes and what it combining with. The first part in column B, P5P, P6P, P7P adopt primary fate, P4P and P8P adopt secondary fate. This is the answer of it. Now there are more cases which we can see. Here I have written directly what is the answer, in front of that only answer is written by decoding it. Loss of function of lin3, all VPC adopt tertiary fate. Loss of function of lin3 and gain of function of 23, but is formed multivulva formation take place. And C part combined with third, reduced function of lin3, P6P adopt primary fate, P4P, P8P adopt secondary fate. Overexpression of lin3, D is going with one part. P5P, P6P, P7P adopt primary fate, P4P, P8P adopt secondary fate. Now we'll see certain other cases which can be seen in the exams of CSIR net, conditions and the results of it. If low lin3 or low Let23 is there, no primary VPCs will be formed. If anchor cell is destroyed or ablated or removed, all 6 VPCs will form epidermal and hypodermal tissue. No vulva formation will take place. The third case, which is written with green color, if lin3 blocked or mutated, no primary VPCs will be formed, no Let23 will be formed, no vulva formation will take place. Fourth part written with red color, if Let23 blocked, no vulva formation take place in C.elegans. If gonad cells destroyed, vulva form as lin3 is present in anchor cell. Tertiary VPC is destroyed if vulva will be formed. Seventh case, anchor cells position shifted. If anchor cell position is shifted, which is just under the gonads of the C.elegans, vulva formation position get changed. If 3 central VPCs are destroyed, rest of 3 cells will form vulva that was previously forming the skin, hypodermis or epidermis. Like this, we have discussed 2 questions fully and the conditions and results of the C.elegans when something is ablated, something is destroyed, mutated, or blocked. See children, everything is possible in the life. Nothing is difficult in the life. Look and see the video to understand again and again, to clear the concept. Friends, like the video, subscribe the video, press the bell icon, and have a happy time. Stay blessed in this time of hard with the word.